Hello, I'm Pedro Antonio Valdes-Sosa. I'm one of the co-chairmen of the Global Brain Consortium, and I am the head of the laboratory that's a joint Cuba-China laboratory of neurotechnology at the University of Electronic Science and Technology. And I'm also a researcher and founder of the Cuban Center for Neuroscience. So I'm going to be giving an introduction to qEEG. I want to first make a disclaimer. qEEG is quantitative EEG. Therefore, in theory, I should be explaining many new developments, but that include linear, nonlinear, time domain, frequency domain methods. But I'm going to restrict myself to the subset of quantitative EEG methods that, one, have some clinical application that has been demonstrated, and two, which are on the portal of C-Brain. As has been, this is the venue for the work of the Global Brain Consortium, and it's been demonstrated how to use this portal as well as EEG-NET in previous talks of this lecture day. I have no disclosures to reveal, and therefore with not much more ado, I will continue my presentation. There's a special issue of Neuroimage called Neuroimaging in Global Health. In the editorial, which was written by Maria Luisa Bringas, who follows me in this series of presentations, we recount how in 2016 the World Health Organization and the Organization for Human Brain Mapping organized at the headquarters of WHO in Geneva the meeting of all the global brain projects to analyze what was the impact of all these brain projects, all this money expended in research on the global burden of brain disorders. Now, basically the conclusion is that much had to be done. Among these, one of the problems was the lack of translational bridges between high-level neuroimaging and population brain health. So that is why we created in 2019 the Global Brain Consortium, as Alan has told, and we focused our attention on the modality that could serve as this translational bridge. This diagram shows the sequence of events after one has neural activation. And each of these steps or stages in the events that accompany synaptic transmission, potentials in the neurons, be they postsynaptic potentials or action potentials, the consequences of this activity, which requires ATP and therefore glucose and oxygen and involves the astrocytes, and later the sequence of signals that occur and produces changes in cerebral blood flow, volume, and produce ultimately the bold signal. So with neurotransmission and activation, neural activity, energy consumption and metabolism, hemodynamic response, we have a sequence associated of different types of imaging modalities. These are all based on a good knowledge of the anatomy. That's the common reference, and we picked EEG, not even MEG, but EEG as the possible translational bridge due to its low cost, its easy deployability, and the experience that has been obtained to be able to use it in many settings. Now, it is very significant that we're having this symposium and also this lecture on the 100th anniversary of the discovery by Hans Berger of the electroencephalogram, EEG. Faisal Mushtaq, who's at the conference, has done incredible work coordinating different groups. You can look at much of this work at the website that you're seeing up on the top of the slide, and a sequence of events that start with a public demonstration and recreation of what Hans Berger did in Jena will be followed by a article that's soon to be accepted. It's in the final stages in Nature Human Behavior, with a survey of current opinion about what is the present and future of EEG. Then, finally, a Belt and Road conference in Chengdu on population brain health that will also celebrate the 100 years of the discovery of EEG. And finally, a EEG manifesto launch by the Cutting Edge group that will be in Berlin in the latter part of the year. So it's very significant that we're at this stage, and I want to get now into what we call quantitative EEG in the narrow sense that we're looking at frequency, domain descriptors. We know that this is not sufficient, but it's a start, as I said. So structural and functional descriptive parameters, we'll call them DPs. We can look at and chart brain developmental trajectories. Now, for this, what we need is to extract from the brain these descriptive parameters, and then in a large group of subjects, even better with longitudinal studies, which are not that frequent, and even with cross-sectional studies, see the dependence of the descriptive parameter on a clinically relevant covariate, let's say age. And once we have this and can describe the probabilistic distribution, both of the mean, the variance, perhaps some other, we can then assign probabilities of a subject belonging to the normative data set or not. So a probabilistic distance, which is based on eliminating as co-founders things like age, sex, and we'll see that there are others. So, recently there was a paper in which many of us collaborated. It's a Nature paper. It was headed by Seidlitz in Bethlehem, and it pooled data across the world for MRI recordings. And you see here the distribution of countries, and it corrected for differences in countries and so forth. And there's now charts of the volume of the brain in this open site on the web, which allows one to compare a given individual and see how he is with respect to the average brain development. This has created a lot of excitement and citations. However, I have to mention that in the history of the EEG, which we're celebrating once again, this approach of looking and doing developmental brain charts was essentially quite old. Colleagues of ours, collaborators, Matushik and Pedersen, already in 1973 described the concept of brain age. And normative modeling, the type I've described, was the work of the Cuban group in what now is the Cuban Center for Neuroscience and the Brain Research Labs of New York. In 1977, in Science, we came out with a paper where we called it neurometrics. That's to say, use EEG to measure directly the activity of the brain, create a probabilistic subspace of normal development, and then try to see what are the profiles of pathology that occur with respect to this the normative subspace. And many of the issues that are now being addressed were already looked at in this paper. In Cuba, we have a long history of this. We introduced quantitative EEG in the public health system in the early 90s with Cuban equipment. We created what was called a high-resolution quantitative EEG analysis. That's to say, to look at narrow frequency bins instead of the broad bands that are used in clinical science. And then we came up with high-resolution spectral norms. We see on the right, on the top, one of these norms, one of this normative data from the first wave of the Cuban Human Brain Mapping Project, in which we see in the axis, vertical is log of the spectrum. The x-axis would be the frequency of the EEG components, and let's say the y-axis would be age from 5 to 90. This was a random sample from the general population in collaboration with the Cuban family doctors. And, you know, in Cuba, studies of this sort have 95% compliance without any type of reward. Now, what we have there is the mean of the spectrum for a given derivation as a function, as I said, of frequency and age. And what you see there, that's the ridge that starts in very high theta, then goes to alpha, then goes back a bit, is the alpha rhythm. That's the description of the alpha rhythm. And you see below how these curves, cross-section of this, are shown, and you can see the speed up of the basic rhythm in childhood to alpha maturity, and then a decrease again with age. Now, we started testing these norms, and in fact, the reason for creating the Cuban Normative Project in 1988 was a health problem. And we created a network of Cuban qEEG systems administered by the Public Health Ministry. And this is how we created the first norms, okay? From a sample of 116,000 subjects, we randomly selected an age and sex stratified sample. We digitized the information of these participants anonymized. Then we did a random selection. And then the family doctor screened the subjects very stringently, and then we went on to record using what we call the Medicid 3M system, which was part of the line of systems that we developed since the 1969-1970 in Cuba, and we created the first Cuban normative database. These norms were then tested, as we'll show later, in a network of clinical neurophysiology labs that were created by our center in the Ministry of Public Health. You see on the left a 128-channel system from 2002. It was a very early model that Maria will later explain how we used it in studies of the presenilin 1 mutation in Alzheimer's in Colombia. And then on the right, you see the modern family of Cuban equipment. Now, what is what we call classical quantitative EEG? This is simply using the log spectra and their Z transforms. Let me unpack this. So we have, if we go from the top left, we have the recording of the EEG at least the 1020 system, 19 electrodes. We do the Fourier transform. In the middle, we see what's known as the cross-spectral matrix. This is a tensor. That means it's a multi-way object that has the dimensions of channels, number of channels by number of channels by number of frequencies. Now, if one picks out the diagonal, which is a real number, then one has the spectrum, and actually we see this for one channel, which is an occipital channel on the head, and you see there the spectrum plotted against frequency, and you see the peak of the alpha rhythm. This is in one subject. You map it topographically, and you see the topographic map, and it's concentrated on the back, as all clinical neurophysiologists know. Now, we can take any of these spectral values, and then it was shown very early on by the joint work. This is a paper of our lab with New York University, and here you see a paper of Roy John and Leslie Pritchett, where they show that you increase the specificity of the norms if you include the confounder of age. If you don't take it into consideration, it's unwanted variance. To achieve probabilistic statements, what we do then is to do the log transformation. And there are different transformations for different parts of the cross spectrum and different parameters. But in this case, it's the log spectrum that achieves an approximate Gaussianity. And therefore, when we do the Z transform, which is taking any descriptive parameter, taking out, subtracting the mean according to the age, and dividing by the standard deviation according to the age, we have a Z value, which if we have a approximately normal distribution, we can assign probabilities of that measure belonging to the normative set. Briefly summarized pictorically, we have different electrodes. We amplify the difference with respect to a reference. We have the recording on the right. We obtain the descriptive parameters. We then transform them, as I said, and then we can have a characterization of normal brain development. But then we can also measure with the Z-transform the deviations from normality. Now, this high-resolution quantitative EEG, you see the papers there where we described it, essentially are based, as you see down there, on the Fourier transform. We then calculate the cross spectrum. This is the covariance matrix at each frequency of the Fourier coefficients for different segments of EEG. These are the samples, no? And this leads at one given frequency to what is known as the cross spectrum, which is a Hermitian matrix that has complex values. And on the diagonal, we have real numbers, which are the spectrum, as I say, if you go down over the frequencies. Now you see this is a box of slides. This is just one slide, what I'm showing you here. If you look at the complete stack of slides at one point of the diagonal, that's the spectrum. If you look outside of the diagonal, that's to say off-diagonal elements, then you have what is a covariance. It's a complex covariance. And this is known as the cross spectrum. And this shows differences, both in average phase and, a correlation of amplitudes. And this can be normalized to the coherence. So basically, this is the basic element that goes into the analysis. Then, when we stack all these cross spectra together for all the frequencies, we end up with the tensor I was mentioning briefly before. Many people concentrate only on the diagonal, and that's what we do in classical qEEG. But we'll see later that we shouldn't ignore the cross spectra, which some people put in the category of functional connectivity, a term I hate, because a correlation, a cross spectrum, is not a connectivity. It's just a correlation. Connectivity is something different. Anyway, for later discussions. So the spectrum, you see here, this was the example I was giving before. And now, let's say we take that value of the spectrum. And then we see here from our data how we carry out heteroscedastic, that is a standard deviation varying regression of both the mean and the standard deviation of that parameter. This is, for example, the peak, around the peak of 10 hertz, right? And we see how it changes with age. With that, we can construct an approximate normal distribution and do the Z-transform. And that's why we need, as you see on the bottom left, both the mean, which is what we're showing here, a regression function is a function of frequency and age, and also the standard deviation. Now, this is at the scalp. We know, and we see here in the diagram, we have, let's say, 3 sources, and they're all mixed due to volume conduction. The solution to this is the area of electrophysiological source imaging. We know that if we do a quasi-static approximation of Maxwell's equations for electromagnetic fields, we can have a description of the different current sources produced by the difference, the currents created by concentration of postsynaptic potentials in pyramidal cells. And what will happen is that each source is reflected in each electrode linearly. And these coefficients, these linear coefficients, when we stack them together as a matrix, is known as the lead field, as we show there. And the general equations in the bottom, and finding, stating the EEG source problem as this way, where we have observed voltages, we have then this matrix of lead fields, we have then currents plus sensor noise. Well, that is the forward problem, okay? Then we must solve this problem, and that is the inverse solutions, of which there are many, and I won't go into much detail here. I will mention, though, that we developed in 2001 one of such solutions, the variable resolution electrical tomography. This is in the 2001 paper by Jorge Bosch, in which we state statistical parametric mapping of sources of the EEG spectrum. And here then we add as a set of descriptive parameters the EEG sources. Now, when you use variable resolution electrical tomography to describe the maturation of EEG sources, well, here this is from 5 to 80 years, as I had mentioned before. And what you see for each voxel, you see what we call the developmental surface. That is to say, the same thing as for any electrode, but now in the sources. And well, that's the paper which Carl Friston described as the first application of statistical parametric mapping to electrophysiology in his handbook on SPM. Now, what can we use this for? Imagine we do a source image with Barrett, and that's what we have on the left of the numerator. We then can subtract the age corrected mean value, and then we can divide each voxel by the age corrected standard deviation. And if you look at the left part of the numerator, and then on the left at the Z transformed image is what we call it, you can see it's totally different. In fact, what we found is that to a certain degree the amplitudes are determined by how close the sources are to the electrodes, whereas in the Z image this influence disappears. So we have not only a deviation in an image from the normative values for that age, but we also have a correction for the electrode bias of source imaging. Okay, so what we wanted to do now is to find the correlation of all these measures with other types of measures. So we carried out the second wave of the Cuban Human Brain Mapping Project. This was published in Scientific Data. All the data and the programs are available, as will be explained later by Bosch. And here we once again sampled from the general population, stratified by age and sex and other demographic parameters. We obtained blood for future DNA studies. We did the EEG. You see there a modern version of our 128 channel system. And at this time we were able to do MRI with 1.5 Tesla. This was a couple of decades ago, before Rainer and Camille helped us get the old Maastricht 3 Tesla system. And there you see the samples that we did of other pathology. So we have a multimodal database, and with cognitive and clinical data, that's what's available as open source. Now, what was the other change besides comparing with other modalities? That instead of limiting ourselves, as we did and we show there on the top left, and I explained before, to the diagonal, to the spectra, which is simply a measure of the energy at each electrode, we now looked at the off-diagonal elements, at all the spectra and cross spectra. And this allows us to look not only at the energy at each frequency at each electrode, but what is the shared variance, what is the coupling between electrodes. I have to say that this has been well known. The spectrum has an approximately distribution that can be approximately, made Gaussian with a logarithm. But then you can take any element, any of those tubes that go along frequency at any of the off-diagonal elements, and split it up into coherence, which is the complex, it's the modulus of the equivalent of the complex correlation coefficient between two electrodes, and then you can find the phase, since this is since cross spectra are complex numbers. And here you see two examples from, so the spectrum is from the occipital region, and the other's occipito-frontal connections, and you can see that there's a spectrum, and there's also there's a coherence, that peaks at alpha, and then you have phase differences. Now, the point is that this object can be transformed as we want to obtain univariate Gaussianity, but the object itself, let's say this tensor, this whole number of derivations times number of derivations times frequencies, that box of slides that I was mentioning, no, where each slide is a cross spectrum, that object does not reside in Euclidean space. We can't apply ordinary multivariate statistics. In fact, I can illustrate that very simply, and here we go. This is an example from a covariance matrix that you see there. It's just a two-dimensional covariance matrix. You have the variance of one variable, you have the variance of the other, and then the off-diagonal, the covariances. If you plot these in 3 dimensions, where you have variance in the x, variance in the y's, and then you have covariance in the z's, the fact is that the condition that this matrix be positive definite restricts the possible values of that covariance matrix to what's known as the semi-definite cone. And here you see it. It can have, let's say, it can be flipped in phase. Now, what happens with this type of let's say, geometry, which is known as a manifold, and when data lie on a specific non-Euclidean manifold, you just can't go about doing averaging or regression. Here we constructed an example where we have data that should actually, when you average them, since they're symmetric, above and below, it should be at 0. That would be the, let's say, the Riemannian. That's what these are called, Riemannian manifolds. But that would be in 0. And however, if you just use Euclidean statistics, ordinary statistics, even the average falls out of the 2 cones. It's not a semi-definite, it's not a covariance matrix. So one has to use other ways of averaging that take into consideration the shortest path on the manifold. In this case, it's called the geodesic. And this is Riemannian, this is statistics and Riemannian manifolds. Now, having this in place, that we need to do correct statistics, which we introduced, we then decided to make a call through the GBC to carry out an extension of the Cuban Human Brain Mapping Project for cross spectra, and we involved a multinational norms project which is still going on. Here we see on the right top the countries, which were 9 countries. We had not only different countries but different systems within each country, because we were interested in seeing the effect of system. Essentially, we did not. Some people had shared the raw data, but others didn't. So we standardized on just sharing the cross spectrum. This is a type of quasi-federated learning. We did quality control on these cross spectra, and this allowed us to carry out this multinational study. We then did pre-processing, and we eliminated a series of bias. I won't go into detail. This is a paper by Ming Li that came out in Neuroimage, in the same issue that I mentioned about neuroimaging and global health. And then we did automatic quality control. And for this, we did TSNE, which is a manifold mapping method that allowed us, for example, you can see there, we could pick out groups that were outliers. We even found one site that had sent us the data, and they were out there in that cloud because they had placed the electrodes on the head in a different order than everybody else. We also use the so-called DD plots, which are a comparison of a robust Mahalanobis distance versus the actual one. And of course, we did visual quality control at a first level to see if anything was wrong. This was pretty effective without looking at the raw data. Now, one of the questions we went into is whether there were batch effects. And batch effects came up in molecular biology because the same system over time could have a drift. You can even see there some examples of changes across sites for cortical thickness, since this idea has extended also to morphometry. You see other examples for diffusion tensor imaging, and the impact of batches, if they exist, which is undesired heterogeneity due to different procedures, due to different equipment, due to the drift of the equipment, or even to characteristics of the people, is that the results are poorly reproducible and they're not reliable. And this is an obstacle to use data from one place in another place. So what we did was to determine batches as a combination of country and equipment. I won't go into details. We did the statistics. This is non-parametric statistics for the mean and the standard deviation. And we include as terms possible batch effects, and indeed we found these batch effects. So therefore, when we're talking about developmental surfaces, we're talking in this context, having eliminated the batch effects. That's why we call this project Harmonize qEEG. In fact, the full name is Harmonize Multinational qEEG, or HARM-MN qEEG. Now, here's an example of the elimination of the batch effects. These are the statistical tests. You see up on the right, which I think is the clearest, you see that there's clusters of outliers. Once we harmonize, these disappear, becomes homogeneous. We see then on the left the Z transforms that are obtained before and after normalization, and indeed the assumptions of Gaussianity with the same mean and standard deviation across all batches is sustained. Having this in hand, we were able to obtain the norms, and here we see a representation. What is so striking here is that these norms, multinational batch-corrected norms, let's say for occipital and parietal and top-bottom, have the same characteristics as those described for the Cuban database of the 88-90 when we published these first results, which shows that the EEG is indeed a very stable result. It's very robust. And then we can break it down. It's in the paper. You can see it either at a given frequency across age on the right, or you can see curves that show you how it varies, how the average spectrum from a given derivation changes with age. Now, the interesting thing here is that since we're using a Riemannian framework, we're now able to do these regressions for the full cross-spectral matrix. As a consequence, we're now able to exhibit as results for phase, for coherence and phase, top coherence, phase down, for parieto-occipital, for example, and these were obtained simultaneously with the other in the same statistical framework. So we have these norms, so we have the standard deviations, we have the means corrected by age, and this is a new result that we have. So basically, these are the ideas. You will see that as the examples that Maria will explain and the practical demonstrations shown by Jorge Bosch, all this mathematics has a particular effect. First, with classical qEEG, we're able to obtain for many years consistent results in discriminating development and disease. But with this new procedure, which we call harmonized multinational qEEG, we obtain even stronger results. But that will be part of the next presentations. Thank you very much for listening to this presentation.